Hello everyone, this is your educator Khushbu and today we are going to discuss on your first chapter, that is the heredity and evolution. In the chapter, one topic is there, that is the transcription, translation and translocation. So first start with the what is transcription, what is translation. Okay, so cell. hmm, Inside the cell nucleus is present, and in that nucleus, our genetic material are present, and those genetic material are DNA. DNA is the genetic material in most of the organisms except some viruses. In some viruses, RNA is genetic material. DNA carry information, carry characters from parents to their offspring. And in DNA, some informations are present in the form of coding, and those informations are used to synthesize the protein. And that pathway is known as the central dogma. Central. So, what is central dogma? The information which is present in DNA is utilized for the formation of protein, and the complete pathway is known as the central dogma. So, when information present in DNA is utilized for the formation of RNA, this process is known as the transcription. And then there is the synthesis of proteins from RNA. This process is known as the translation. One more concept is there which is not mentioned in your book, that is the replication. DNA has the capability that it can make its own copy, and this process is known as the replication. So when we are talking about the central dogma, so central dogma is a pathway and it consists of three different processes or three different steps. When DNA makes its own copy, the process is known as the replication. बिकॉज क्या होता है, बिफोर सेल डिवीजन ये जो भी जेनेटिक मटेरियल प्रेजेंट है उनको क्या करना पड़ता है, उनको अपने नंबर को मतलब डीएनए को अपने नंबर को इंक्रीज करना होता है बिफोर सेल डिवीजन, जिसकी वजह से रेप्लिकेशन प्रोसेस जो है ये डीएनए परफॉर्म करती है. अभी आफ्टर दैट व्हेन व्हाट आर द इंफॉर्मेशन प्रेजेंट इन डीएनए, by using that information, there is the formation of RNA. उसे हम क्या बोलेंगे, ट्रांसक्रिप्शन. एंड फ्रॉम आरएनए टू प्रोटीन हम बोलेंगे ट्रांसलेशन. और ये जो पूरा कंप्लीट पाथवे है ना, वो होगा सेंट्रल डॉग्मा. यानी कि सेंट्रल, द मीनिंग ऑफ सेंट्रल डॉग्मा इज डीएनए टू आरएनए, आरएनए टू प्रोटीन, डीएनए टू आरएनए इज ट्रांसक्रिप्शन, आरएनए टू प्रोटीन इज ट्रांसलेशन, एंड डीएनए टू डीएनए इज रेप्लिकेशन. Next is what is DNA and what is RNA. As I told you that DNA is the genetic material in most of the organisms and RNA are genetic material in some viruses. So when we are talking about the chemical composition of DNA, so DNA and RNA का जो chemical composition है, that is almost similar, but certain differences are present there. So DNA is mostly consist of three different components. First is your nitrogenous base, and second is your phosphate group, and third one is your sugar. तो डीएनए और आरएनए का जो डिफरेंस है ना, वो इस टू कंपोनेंट्स में अराइज होता है, नाइट्रोजनियस बेस और सुगर में. So when we are talking about the nitrogenous base of DNA, so DNA में 4 टाइप के nitrogenous base present होते हैं, adenine, thymine, cytosine and guanine. Adenine is always paired with thymine with double hydrogen bond, and guanine is always paired with cytosine with triple hydrogen bond in case of DNA. But in case of RNA, RNA. Thymine is not present. It is replaced with the uracil. So this is one of the important difference between DNA and RNA, that in DNA, adenine, guanine, cytosine, and thymine, 4 types of nitrogenous bases are present, but in RNA, thymine is replaced with uracil. Differences in case of sugar, right? As I told you, see, maltose sugar, which is present, which is a chemical component of DNA. So this sugar is consist of 5 carbon, 1, 2, 3, 4, and 5. So as you can see that in case of DNA, the carbon number 2, right, 1, 2, here, this is hydrogen group is present, but in case of RNA, at carbon number 2, hydroxyl group is present. Means in DNA, ऑक्सीजन ग्रुप जो है वो क्या है, रिमूव है. दैट्स वाई डीएनए का जो नेम है दैट इज द डीऑक्सीराइबोज, मीन्स डिराइवेटिव ऑफ ऑक्सीजन ग्रुप. बट आरएनए में सेकेंड कार्बन जो है वहां पर क्या प्रेजेंट होता है, हाइड्रोक्सिल ग्रुप प्रेजेंट होता है. तो डिफरेंस बिटवीन डीएनए एंड आरएनए that is generally asked in your exam and 2 to 3 marks का क्वेश्चन होता है. तो अगर आपको ये क्वेश्चन आता है एग्जाम में कि लाइक व्हाट इज द डिफरेंस बिटवीन डीएनए एंड आरएनए, तो आपको ये 3 पॉइंट्स कवर करने हैं. फर्स्ट आपको ये लिखना है कि डीएनए इज अ डबल स्ट्रैंडेड कंपोनेंट बट आरएनए इज अ सिंगल स्ट्रैंडेड कंपोनेंट. In case of DNA, 4 types of nitrogenous base are present: adenine, thymine, guanine, and cytosine. In case of RNA, thymine is replaced with the uracil. Okay, and in case of DNA, the जो second आपका carbon है, that is the derivative of oxygen. At the second carbon, hydrogen group is attached, but in case of RNA, with the 2' carbon, hydroxyl group is attached. Fine. Okay. So this is your difference between DNA and RNA. And hope what is central dogma that is clear for all of you. अगर आपके कुछ भी questions है, कुछ भी doubt है, you can ask me in comment box and I will try to resolve your question. And if you like the video, please subscribe the channel. Thank you.